Hello, this is our final lecture on recreational drugs. A reminder that nobody associated with the course condones or advocates the use of recreational drugs, but that I do drink coffee and tea. First, I'll tell you about dopaminergic neurons. We certainly have mentioned the dopaminergic system before. In fact, you may remember that in the lecture on nicotinic acetylcholine receptors as molecules, I showed you the handlebar mustache. The handlebar mustache was a coronal view, and this is a sagittal view. The most important players in recreational use of the dopaminergic system are the ventral tegmental area, which was the upper lip of the handlebar mustache. The ventral tegmental area projects to many areas of the brain, releasing dopamine in many areas of the brain. For the sense of pleasure and well-being, the nucleus accumbens is the most important projection of the dopaminergic neurons. It is also called the ventral striatum. The other part of the handlebar mustache, the handlebars themselves, or the substantia nigra, it projects mostly in humans to the dorsal striatum, which mostly concerns movement. Although in humans, there is pretty good indication that even dopaminergic neurons in the substantia nigra projecting to the striatum are associated with feelings of reward or well-being. Now we turn to noradrenaline and to the noradrenaline system. The noradrenaline system also projects to many areas of the brain, as well as to descending areas in the spinal cord. Only a few thousand neurons in the brain make noradrenaline. They are mostly in a place called the locus ceruleus, which means literally blue place, because in some stains, the locus ceruleus neurons are indeed blue. In terms of system-level action, drugs that affect the dopamine so-called pleasure system include morphine and heroin, nicotine, cocaine, amphetamine, perhaps ethanol, caffeine, and phencyclidine. So many drugs associated with recreational effects affect the dopamine pleasure system. There is a lovely circuit diagram of the cells in this pleasure system, and I hope to get to it in a future lecture. The noradrenaline readiness system, dominated by the locus ceruleus, is also affected by nicotine, probably by cocaine, by amphetamine, and by caffeine. Now there are two other systems, one that I call the perception association system, and one that I call decreased neuronal activity, that are affected by other drugs. The perception association system is affected by tetrahydrocannabinol, by LSD, and by PCP. And decreased neuronal activity seems to be a general effect of morphine and heroin, of THC, of ethanol, of LSD, in fact we'll see this in just a few slides, and of PCP. Looking at these final two columns here, let's talk about the serotonin neurons. You've seen a diagram of the raphe in a previous lecture discussing serotonin. That was a rodent raphe. Here is a human brain with the raphe nuclei clearly marked. They also project to many areas of the brain, including ones we've discussed previously, as well as to the spinal cord. How it is that selective actions on serotonin neurons cause decreased depressive symptoms when those drugs are inhibitory, and hallucinations if those drugs are excitatory, is not yet clear. In fact, let's test our knowledge of the serotonin system with a journal club. as though we were in a university laboratory discussing the latest papers. Here's one from 2012. This paper used psilocybin, which the body converts to psilocin from mushrooms. The paper used functional magnetic resonance imaging, abbreviated fMRI. The local capillaries expand, producing greater blood flow, and the fMRI registers this. First of all, though, let's talk about how the subjects in this experiment felt. There were 15 human subjects. They were given either psilocybin during an infusion that lasted about a minute, or placebo. And in a good experiment, neither the experimenter nor the subject know what's in the syringe. The subjects clearly reported changes in consciousness. They reported much more observations such as, I saw my surroundings change in unusual ways. I felt unusual body sensations. Sounds influenced things I saw, so you can read these captions for yourself. So the subjects certainly were having a trip. And let's see now what the fMRI showed during these infusions of psilocybin. Here's the left hemisphere, here's the right hemisphere, the midline. The surprise was that actually there were no brain regions activated, but many brain regions received lower blood flow as though they were working less hard. So here is the color code. This lower blood flow occurred in the anterior cingulate gyrus, the posterior cingulate gyrus, and also in the thalamus. Again, there were no observations of increased blood flow, no observations of the overall set of neurons working harder. Mostly they were working less hard. Now, if we look at the time course of the changes in blood flow, for the placebo, there was actually a bit of an increase in neurons working harder. But in all of the three regions we pointed out, we see a lasting decrease in blood flow. So here's the infusion, which lasted a minute. This is before the infusion. This is after the infusion. The experiment went on to about 18 minutes. And clearly after the infusion, while the psilocybin was present and was being converted to psilocin, the overall effect is that neurons seem to be working less hard, presumably firing fewer action potentials. Well, the investigators point out this surprising result. In fact, the regions that were most active before psilocybin showed the greatest decrease during the drug. The authors were able to show that a few papers in the literature could have foreshadowed this result, and they suggest that psilocin activates some GABA neurons. These are inhibitory neurons, and this would decrease the overall activity of the more numerous glutamatergic neurons. So you can see that there are still many, many surprises in drugs and the brain, and we can expect more surprises like this in the coming years. Overall, then, we can say that recreational drugs have varying effects. Morphine and heroin are primarily inhibitory. Tetrahydrocannabinol is primarily inhibitory. Ethanol is primarily inhibitory. On the other hand, some recreational drugs have overall excitatory effects. Nicotine, cocaine, amphetamine and derivatives. But I do want to remind you about the paradoxical use of amphetamines for ADD, ADHD. They apparently calm down people. And caffeine are excitatory. And then there are the drugs that we can't classify as overall inhibitory or excitatory. We say that they produce hallucinations. Finally, let's discuss the legal status of recreational drugs in the USA, at least in late 2012. Morphine and heroin for recreational use are illegal. For prescription use, they are in Schedule 2 for pain control. There is no known non-prescription use. For tetrahydrocannabinol is illegal for recreational use. For prescription use, the situation changes every day, and you can summarize it for yourself by looking at the newspaper. Nicotine is taxed, sales to minors are prohibited, and it is possible to get a prescription for a smoking cessation product, but you don't need a prescription for a smoking cessation product. And smoking cessation uses of nicotine include patches, gums, and inhalers. Cocaine is illegal for recreational use. It is used for some ear, nose, and throat surgery, very delicate surgery. Amphetamine and derivatives are illegal for recreational use. For ADD and ADHD, they are on Schedule 2. And there are some diet pills that contain amphetamine and derivatives available without prescriptions. Ethanol is taxed. Sales to minors are illegal. I know of no prescription use of ethanol. LSD is illegal and has no prescription use. Caffeine is legal. For prescription use, it's still in some migraine medications, and over the counter it's possible to buy antihistamines that have caffeine in them. Phencyclidine is illegal and has no current prescription use. For the next few mini lectures, we're going to talk about nicotine addiction.